Folks, without further ado, thank you very much. I'm grateful for the fact that you all went to the trouble of being here at this rather early hour. I wanted to show you a few pictures by way of a sort of a wake-up mental exercise. Oh, you're not waving to me. Hi. So this, of course, is Sir Isaac Newton. It is his statue in Trinity College in Cambridge, where he, according to Apocrypha, sat under a tree, and then an apple fell on his head, and then he realized that gravity is actually a tangible phenomenon. Something that we all observe on a daily basis. You're not floating off your chairs, although I'm sure some of you wish you were, because of gravity. Another truism, as you walk out today, you'll look up and you will see that the sky is blue. Last but not least, something that's accessible to all of us, that's part of our intuitive, immediate experience, is heredity. I don't suppose any of you know who this is. By show of hands, somebody? That's Kirk Douglas, the great American actor. And that's Michael Douglas, his son. Now, is acting inheritable? In other words, is there an acting gene? Nobody knows. But there's certainly a facial features set of genes, as you can see here. You will say to me, why are you telling us this? It's 8:10 in the morning. Well, because it's interesting. As I'm about to explain, gravity is in fact not understood. The blueness of the sky is understood, but highly technical. On the other hand, heredity is understood and is not technical, and is the subject of this section of the class. Specifically, why did the apple allegedly fall on Newton's head? In fact, technically speaking, nobody knows. There is not a sustained conjunct mathematical model or physical model of the function of the gravitational field. The sky is blue because, I don't want to screw this up, the extent of inelastic Rayleigh scattering is inversely proportional to the wavelength of light being scattered raised to the fourth power. And thus, if you parse the input histogram of sunlight through that equation, you get the blue sky. Ugh. On the other hand, children look like parents because Mother Nature has evolved a thing called DNA. It's the basis of heredity, and evolution has shaped DNA in a way that it is eminently copyable, such that the instructions for facial features could be passed on from Kirk Douglas to Michael Douglas and from parents to children the world over. So today I will tell you a little bit about the fact that, as all of you know, but I'm hopeful we'll rediscover today in a potentially new angle for you, DNA is the genetic material. It has a structure that is beautiful. It replicates semi-conservatively, and I'll explain what that means. It has nothing to do with its political beliefs. It has to do with biochemistry. And it can be amplified by the polymerase chain reaction. First and foremost, let us stare at the DNA itself. So I want to emphasize that while I'm about to show you is first an image, then a movie, this is a very strange movie. It's an atomic resolution reality known as a crystal structure. And a core part of our exercise today and on Friday will be to understand what crystal structures are and why they're useful. But without any further ado, folks, I give you, I hope this works, this better work, the crystal structure of DNA, and let's play a movie of it. If this doesn't work, I apologize. All right. Movie, program, camera loop, why rock? Why rock? Because it's great. Why roll? There we go. And now, play. And absolutely nothing happens. On tonight's episode of Violently Anticlimactic, I'll just rotate it. Oh, there we go. I'll do this. So this is DNA face like this. You see its beautiful minor groove and major groove, these two strands woven around each other. One thing that's interesting about DNA is what it looks like from the side, and that looks like this. I know. Believe me, I've been looking at this for 30 years, and I still get that feeling. Whoa. These are just pretty pictures. What do they tell us about how we are put together? Well, that's the subject of today. Bad movie, bad movie. Why didn't you play? You played at home at 5 a.m. this morning. You will say to me, what is 1 Bravo Nancy Alpha? It is an access code for that image. And those of you who are interested, write me an email, and I'll tell you how to access that image on your own computer. So you can have your favorite website on one side and the DNA structure on the other, and see who stays your friends on Facebook. I felt obliged to connect this structure discovered in 1953 to events of February 2012. And recent history offered a marvelous opportunity to do so. It has to do with the use of DNA to cure blindness. And when I say cure blindness, I actually mean cure blindness. 20% of children in schools for the blind have a genetic condition called Leber's congenital amaurosis. Will this be on the midterm? No. There is no treatment for LCA, that's the acronym for the disease, and severe visual impairment during childhood progresses to total blindness by the 3rd or 4th decade of life. So by way of an illustration of a relatively old and a contemporary example of LCA, I'd like to begin with one of my favorite musicians, blind Willie McTell. He influenced tremendously people as diverse as Bob Dylan and the Allman Brothers, and he in fact was nearly completely blind as a child and went completely blind as an adult. February 22nd, 2012, the Daily Telegraph. Twins lose their sight within months of each other. Michael and Dan Smith, 20, are still determined to finish university and get good jobs after being left almost totally blind by Leber's optic neuropathy. Michael, who's in football training for this year's Paralympic Games, lost his sight in a matter of weeks while in his first year at Barts and the London School of Medicine. Less than a year later, his brother Dan suffered the same fate while in his second year studying aeronautical engineering at the University of Bristol. The identical pair, who can only make out shadowy shapes, have been forced to adapt their lives, learning Braille and relearning how to cook and choose clothes. I chose this example of twins for a reason. As I mentioned to you, the disease is genetic. It is caused by a mutation, and we'll learn a lot about mutations in this section of the class. A genetic change in a gene called RPE65. Do you need to know the name of the gene for the midterm? You don't. The product of the gene is a protein required for the normal function of our retina. And what I'd like to do is briefly connect what I'm about to tell you on this slide to what you learned in Professor Malkin's section. You learned from Professor Malkin that, of course, the fundamental act of life on Earth is the absorption of sunlight, of the photon, by the light harvesting complex, this glorious constellation of pigments that take the energy of the photon and pass it on to allow ultimately the biochemical process that was discovered here at Berkeley, which is known as the cycle. Thank you very much. Go Bears, the Calvin cycle. No, it was literally discovered here. I mean, it's one of the more, this university has made many majestic contributions to science, and the Calvin cycle was discovered here. So, just like photosynthesis, vision is a process that begins with the absorption of light. Now, I don't want to misrepresent myself as a physiologist of vision, so I learned this from textbooks just like you are. But I have learned from those textbooks that the photon doesn't just magically enter the visual cortex, which actually happens to be here, and communicates information. It is first absorbed by rods and cones, which are highly specialized cells that sit in the retina, and then they transmit information about the absorption of light via the optic nerve to the visual cortex. The elementary act of vision, and now this I suspect some of you don't know, but I would like you to please know it, that the elementary act of vision is in fact the change in configuration, in the chemical configuration of a one bond in a molecule called retinol. Anybody know what vitamin it derives from? Vitamin A, good for you, vitamin A. So the reason that subclinical deficiency of a vitamin A is bad is it actually leads to defects of vision. About, to the best of my knowledge, 200 million people on Earth So slightly less, but still on a ballpark number, a range of value, close to the number of people that live in this country. People on planet Earth suffer from subclinical vitamin A deficiency. The elementary act of vision, in fact, is once the photon is absorbed, is the change in conformation from cis to trans. Now, the beautiful biochemical cascade that happens downstream of that change and how that ultimately ends up being, by the way, it's poignant, isn't it, that you are seeing this using this. Every photon that hits your retina from that causes this to happen. At an atomic scale, of course. Okay. Once that isomerization happened, the pigment needs to be reset to the ground state. And that resetting is actually affected by dedicated enzymes that live in the retina. Their biochemical function is to perform the reverse reaction, to reset our visual system for being able to perform this. RPE65 is in that enzymatic cascade. And in patients with Leber's congenital amauropathy, that step is blocked. It's tragic, but true. And you saw me show you 2 minutes ago these two twins in England and blind Willie McTell, thousands of people around the world who go irreversibly blind. Their world fades as they go through life. Until 2 years ago, there was nothing one could do for patients with LCA. And then Jean Bennett at CHOP on the East Coast actually cured, and that's a strong word, cure. So I'll tell you what I mean by that in a second. Cured a number of individuals of their blindness by, believe it or not, directly injecting the curative DNA into their eye. And if that sounds slightly sort of bad science fictional, it's not. This was in fact a very sophisticated, multi-year, multi-step clinical trial process. Incidentally, by way of teaching you to fish and thus feeding you for life, before you folks is a screen snapshot of Google for biologists. It's called PubMed. It's just as useful as Google. The only difference is in Google, not every fact is verified by somebody who knows what they're talking about. In striking contrast, in PubMed, the majority of the literature is peer-reviewed, so at least somebody who has some education in this has read this to determine whether or not the statements being made are even close to being true. You just Google the word PubMed and you get to this database of biomedical literature. These two numbers are unique identifiers that will get you to the two primary papers. Do you have to read them? No. Do I recommend that you read them? Absolutely. It's truly remarkable. What I'm going to do is show you two movies. I fought earlier with the sound on the movies and failed, but there's nothing we can do, so I'm going to try to do this. I'm going to try to put the mic itself next to the speaker of the computer and see if it goes. It's not about the audio, it's about the visuals. You will see a boy who I believe at the moment of the movie is 9. He's been essentially completely visually impaired since birth, and the extent of his current visual impairment will be seen by you because he will be asked to navigate a maze with one eye covered. Which eye? The untreated eye first. So this is a clinical trial, right? You need to convince yourself that the treatment actually worked. How do you do that? Well, you treat one eye and you leave the other eye untouched. So in movie number 1, they cover up the treated eye and ask this kid to navigate the maze using the untreated eye. Any questions about what you're about to see? Okay. The kid says, at least I can see the black squares. The kid goes, this is hard. He's that blind. He really can't see a thing. It's kind of painful to watch him. He just stands there with this sign in front of his face. He can feel. He is perfectly fine. He just can't see. Okay? So the physicians then asked him to stop. Opened up the treated eye, the one with DNA injected into it, covered up the untreated eye, and asked him to navigate the maze again. The physicians erupt in applause. They're ecstatic over the child, of course. Can you imagine being the parent of this boy? Having raised this kid to 8 or 9 yearhood and living with a child who cannot even see your face, and then some DNA gets injected into this person's eye, and this happens. My purpose in walking you through this is to say one thing and one thing only, which is DNA. It's powerful. It can have a profound effect on our lives. In fact, it does. We don't really think about it a lot. You don't wake up in the morning and say, I wonder what my genotype says. But we are, to a substantial extent, although not a complete extent, something I'll discuss later, a product of our genes. So my purpose today is, before I can tell you about how this was done, for example, some of you may ask, how did the DNA get into the eye with a needle? Okay. Did it then magically enter the cells? No. It was delivered on a virus. Why a virus? Bear with me and I will explain, but not today. Before we dive into modern-day viral-based cures for Leber's congenital amauropathy, I think it's useful for us to go back in history. Not severely back, to the end of the 19th century, to one of the many examples, and this, by the way, will be a persistent theme in my section of the class. How have biologists throughout the history of science made use of, quote, model systems or model organisms to understand living processes? In other words, at the end of the day, we're all really interested in how humans beings work. But at the other end of the day, it's slightly unethical in an ethical sort of way to do more than very specific kinds of experiments on humans. For this reason, the sea urchin, for example, has been really useful. I'm not pro the harassment of sea urchins, but at the very least, you know, it's just a sea urchin. This is Theodore Boveri. Do you need to remember his name for the midterm? No. Is it good for your erudition in general? I went to Berkeley and I know who Boveri is, type of thing. Yeah. I chose names for the section, not because I like spewing forth names, but because I think it's just nice to know them. And if you say to me, I don't want to remember the names of random people, ask yourself, how many of you remember the last names of every person in the complete cast of Friends? Now ask yourself, why? You will spend the rest of your life with a neuron in your head that remembers the name Jennifer Aniston. I have nothing against her. I don't know anything about her except that there's a person with that name. But why is that neuron in your head? Alternatively, you can spend the rest of your life with a neuron in your head called Theodore Boveri. And Theodore Boveri has actually done a lot more for humanity than anyone who has ever been on TV. Specifically, Boveri proved, believe it or not, a remarkable point, that heredity, this ability for like to beget like, the fact that Michael Douglas looks like Kirk Douglas, heredity lies in the nucleus. I'd like to walk you through this experiment, I'd like you to write it down, and I'd like you to know it. And on the midterm, I'm going to ask you to tell me about Boveri's experiment that proved that heredity lives inside the nucleus. And you will say, what about the mitochondria? In a minute. This beautiful little critter has a lovely Latin name, which you don't need to know, but I just like to say it. Strongylocentrotus purpuratus. Bovary made use of the fact that the egg, the unfertilized egg of the sea urchin, is relatively large, and so it is logistically feasible to remove its nucleus. The technical term that I'd like you to know, because it will then be relevant when we discuss Dolly the sheep, is enucleate. Bovary enucleated a sea urchin egg, an unfertilized one. He then proceeded to fertilize this enucleated egg with the sperm of a different species. First small fact that I'd like you to know about this. If you were to do this experiment with a mammal, so for example, a mouse, it is probably unlikely that this strange creature, an enucleated egg, fertilized by a sperm, would go through even the first embryonic division, where one egg forms two cells. Anybody care to venture an explanation why? The answer is a word that starts with the letter H. Who said that? You're the man. It's haploid. So it only has one set of genetic instructions for the building of an organism. Normal mammalian embryonic development requires a diploid embryo. So 2 sets of instructions. 23 chromosomes from mom, 23 chromosomes from dad, go, baby. 1 set of instructions, 23 chromosomes from dad is like, ugh. Now this doesn't mean that you cannot generate, here are fancy words, gynogenetic and androgenetic embryos. You can, and they look actually really interesting, but they're an exception, not the rule. Okay, sea urchins are not like mammals. If you take a haploid sea urchin embryo, it will develop, which is like, it's Mother Nature doing her thing. Bovary took sperm from species A and fertilized an enucleated egg from species B, and then asked, does the child look like mom or dad? Now, if I ask you this question, you'll be like, of course, dad. But this was 125 years ago. People didn't know that heredity lives in the nucleus. Somebody needed to discover that. Ladies and gentlemen, I give you the input. What are these? This doesn't look like a sea urchin. This is a sea urchin larva. I don't suppose by some miracle, some integrative biology major, or just an animal lover in the room knows what the beautiful Greek word for the sea urchin larva is. Anybody? Close, but no, but you're thinking in the right direction. It's not nymph, it starts with a P. It's Greek for easel, as an artist's easel. Good for you, it's a Pluteus. P-L-U-T-E-U-S. It sounds like a character. I am Pluteus. Here are 2 Plutei of 2 different sea urchin species. This is the species that gave the sperm. That's the shape of its Pluteus. And this is the species that gave the enucleated egg. Note, they are different. That's why the experiment is feasible. If they looked the same, you couldn't determine whether or not the result looks like mom or dad, because mom and dad are identical. Now, I should point out that the next slide will feature a word that in modern-day American English is slightly rude, the word bastard. Back then, it technically meant the offspring of 2 different species. So when we say, the bastard looks like dad, and I cannot do a proper Scottish accent, but I wish I could, because this would be a perfect time to do it. The offspring looked like dad. Bovary knew that the sperm is essentially a nucleus with a tail. Conclusion, heredity lies in the nucleus. What would I like you to know about this? Pretty much everything. I'd like you to be able to write out from a memory the complete experimental sequence. What was the question? What was the hypothesis? What was the experiment? What were the data? What was the interpretation? Yeah. Okay? What inside the nucleus causes heredity? Is it, well, you realize I haven't told you yet what is inside the nucleus. Let me tell you a useful life story. This is actually true. Let's say you want to be a doctor, and I'm hopeful that many of you do, and more power to you. Physicians are an amazing profession. I have one of our physicians at the place where I work is a neurosurgeon. It's a 6 to 8 year residency, and you know how neurosurgeons and vascular surgeons practice their skills? They sew saran wrap with surgical thread until it can hold water. I'm not making this up. And they spend 6 years doing that. Oh. So let's say you want to be a doctor, just like them. But you become hard of hearing. This means you can't even use a stethoscope. You cannot hear how the person's heart beats. You cannot talk to your patient. And you're 23, and your dream of a lifetime is gone, and medicine is no longer an option. What do? Well, you could sulk. You could blame the world, specifically your parents and professors, in particular that guy in Bio 1A, for all this. Or both, 1 and 2. A fourth option is you could find a field of study where your training is useful, get a job with the world's #1 person in that field of study, and make a discovery for the ages. And who did that? The guy who discovered DNA. And you will say, wait a minute, 2 guys discovered DNA. Watson and Crick. James Watson and Francis Crick did not discover DNA. They proposed a model for its 3-dimensional structure. Their model was based on data from a woman named Rosalind Franklin and many other pieces of data. Their model proved to be correct. Remarkably, DNA was discovered over 80 years prior to that by the Swiss chemist Friedrich Miescher. And the life story that I just described to you, this business about going deaf and having to abandon medicine, his life's dream, and then going to Felix Hoppe-Seyler, the leading authority at the time in biochemistry, and then discovering DNA is his life story. He was very interested, Miescher was, in understanding how cells of the immune system work. And he needed lots of them for his experiments. And I know that sounds really gross, but I'm sure this is pre-breakfast for most of you. He actually extracted those cells of the immune system from pus-infused bandages. This was an easily available source of white blood cells. After some 5 years of back-breaking work in the cold room in Hoppe-Seyler's laboratory, Friedrich Miescher discovered a heretofore unknown substance that was exclusively present. And now some large mythical creature will walk through the doorway. Miescher discovered this remarkable substance in the nucleus that nobody had ever seen before, and he had a wonderful predicament, finders keepers. He gets to name it. What do you call that? He called it nuclein. I mean, what else do you call it? You don't know what it does. You don't know what it is. So, subsequently, in a few years, the word nucleic acid was then attached to it, and of course, now we know it as deoxyribonucleic acid, or DNA. It's sad but true. Miescher died at a young age of 51. He died of tuberculosis. Let's see, 52 years before the structure of DNA was proposed. So he did not live to know what he discovered. And in fact, his discovery waited some 80 years to be truly appreciated. So that said, I think, to use an expression Barack Obama likes, teachable moment. The teachable moments from Miescher's life are: when life gives you lemons, study the chemical composition of lemon juice. And you might make a discovery that will resonate through the ages, but be prepared for the fact, as in Miescher's case, look at this. This guy discovered DNA. Can it be better? Well, I don't know. What's better than that? And yet, the time was not there yet for his discovery to resonate. Why? Technology. X-ray crystallography and bacterial transformation, and I will teach you both. were not discovered yet when he found DNA. That said, don't be sad. The fruits of your labors may well resonate but you're no longer around, but don't be sad. There's an institute named after the guy in Basel, the Friedrich Nietzsche Institute for Biomedical Research. So there. What I'm about to tell you is slightly gross is the wrong word, slightly overwhelming, but reality can be tough. By way of telling you about the next chapter in the story of DNA, I need to tell you about necrotizing fasciitis. Necrotizing fasciitis is commonly known as flesh-eating disease. It's an infection of the skin which causes destruction of the underlying tissues and muscle. And by now, you've already learned how to fish. If you go to PubMed, you can pull up these two papers that will teach you about it. About 125,000 people a year die of flesh-eating disease. It is caused by a bacterium called Group A Streptococcus. It also causes toxic shock syndrome. It's not a pleasant bacterium. Recently, and when I say recently, in your lifetime, in the late 80s, there was a remarkable resurgence of necrotizing fasciitis and streptococcal-induced toxic shock. It was clear to bacteriologists around the world that the bacterium had evolved from the relatively benign form in which it existed, basically from the moment of its discovery. And in the 80s, something happened, and suddenly, 100,000 people a year were dying of flesh-eating disease. We now know what happened. The molecular events, I will just read to you a passage from this review that I pulled out. The molecular events involved in the emergence of unusually virulent pathogens are poorly understood, but in this case they are. Bacteria can evolve slowly through the accumulation of point mutations or rapidly in an evolutionary quantum leap by acquisition of new genetic material through horizontal gene transfer, e.g., transformation. In fact, it is known that the flesh-eating disease strain of Streptococcus acquired through transformation a 35,000 base pair chunk of DNA from a different strain of Streptococcus. And this new militant chimera then proceeded to go on this rampage around planet Earth. At this point, there are mental hands being risen in this room. Why are you telling us this? Because I don't want you for a second, when you read the description of Fred Griffith's famous experiments with Streptococcus R and S in mice, I don't want you to, for a second, to think that bacterial transformation is something to be hated with a passion in Bio 1. Bacterial transformation is something that we need to learn to deal with as we face the challenges of 21st century medicine. That said, the fact that Friedrich Miescher's discovery, DNA, is the genetic material. You realize, Boveri showed that heredity is in the nucleus. That was the sea urchin thing. Miescher discovered DNA inside the nucleus. That was this pus-filled bandages thing. Now what? Now you need to prove that DNA is genetic. How do you do that? So I will go through this quickly. Your textbook explains it well. And I'm expecting you to read carefully and understand what figure 14-2 on page 283 of your very expensive and very comprehensive textbook says. In brief, whenever one approaches a problem of such magnitude, DNA is the genetic material. How do you study that? You have to study that using a specific case history. You cannot walk up to Michael Douglas and say, Mr. Douglas, we want to prove that DNA is genetic. Can we have some of your DNA and your father, we know your father, Furthermore, we don't know where in human DNA facial features are encoded. It's just not a tractable system. What experiment are you going to do? In bacteria, however, you can do this. You can rely on the fact that first, Streptococcus this genus, members of which cause flesh-eating disease, also causes lethal disease in mice. Furthermore, that it comes in two flavors, varieties, strains, whatever you want to call them, lethal and benign. Last but not least, get this, if you take the lethal form, kill it, die, and then mix it with the benign form, guess what happens? That's right. One transforms the other. It's like, Glenn Close in Fatal Attraction. It comes out of the bathtub and comes back from the dead. So there's something in the dead bacterium, which is lethal, that communicates to the alive benign bacterium that it must change its ways. Griffiths just discovered the phenomenon that heat-killed lethal strains of Streptococcus can transform alive benign strains of Streptococci into lethal forms. And that set up a beautiful, beautiful experiment by this remarkable gentleman, Oswald Avery, at Columbia. He set out to understand the chemical nature of the transforming principle. And his marvelous experiment, I'd like to teach you today. We have a lot to learn from the way Avery went about his thing. I think that's probably all that I'm going to have time for. My plan is to start at 10 past the hour sharp and let you go at 9 on the hour sharp, because I realize many of you have other commitments. This is the Colgate University Band. This is Avery when he was an undergrad. This is the entire band, folks, in 1900. It's amazing. Times have changed. Go Bears. Avery has entered immortality, and every practicing biologist knows his name and repeats it with reverence because of one study that took 10 years, and that has this long polysyllabic title that just kills mice on sight. It's so boring. Studies on the chemical nature of the substance-inducing transformation of pneumococcal types, induction of transformation by a deoxyribonucleic acid fraction, isolate from pneumococcus type 3. If you make a discovery of this magnitude, entitle it, DNA is the genetic material. Don't give it this title. I'm joking. This is nicely technical. This is a picture from Avery's original paper. You don't have to if you don't want to, but it's so beautifully written. I encourage you in your non-existent spare time, and I respect that you're in college, so you have a lot of time pressures on you. Download this paper and skim through it. It's beautifully written, and it's a monument to the power of the human mind. The biological phenomenon under study is before you. This is the benign, individualistic form of Streptococcus, and this is the evil one. This is the lethal one. Heat-killed extracts of that transform this into this. Fine, so your starting point is a heat-inactivated bacterium transforms a live bacterium. The simplest interpretation, you have to start somewhere. It doesn't pull out a magic wand and says, axio transformation. Something must be happening. There is some entity, a chemical, that causes this transformation. It has a certain, of course there is, but now you need to find it. Avery set out, it took him 10 years, to purify it from the heat-inactivated bacteria. In his own words, our major interest has centered in attempts to isolate the active principle. Nicely old-fashioned English. What does he mean, the active principle? He means the chemical that causes the transformation from crude bacterial extract, and to identify, if possible, its chemical nature. Oh my goodness gracious me, talk about easier said than done. I will show you the biochemical fractionation scheme. Do I need you to know it? No. I will say at the very end of this what I'm expecting you to know. But I do want to give you a sense of the scale of the effort. Step 1, purify the entity. How do you do that? Well, in physics, as you probably know, the algorithm is Richard Feynman's immortal How to Solve Problems in Physics. How many of you know this? Ah, cool, I'll tell you. Step 1, sit down at the table. Step 2, write down problem. Step 3, think very, very hard. Step 4, write down solution. In biochemistry, it's not as easy. The bacterium is an incredibly elaborate mixture of things. And I do want to make one point, and I'll be honest with you, my goal, not for an attosecond, is to indict your textbook, but to simply point out where the representations of reality that it contains are somewhat disconnected from it. If you look at the picture of the bacterium in your textbook, it has sort of a corn dog with stuff, spiky corn dog with stuffing model. It implies that at the heart of the bacterium lies its DNA, clean and neatly packaged. Now, this is convenient from a textbook perspective, but folks, it is simply not true. The DNA of bacteria, and in fact of most cells, is in a complex. It's not neatly packaged, and this is where the DNA lives. No. With a huge amount of protein and lots of other things, and is not neatly packaged away. It's vibrantly chemically active. It directs all the metabolism of the bacterium, and of course it replicates. And to give you a sense of how quickly, I saw a marvelous movie on YouTube from a company called Celasic of E. coli dividing, and I wrote to their chief technology officer, Dr. Philip Lee, who turns out got his PhD here from UC Berkeley. Go Bears. And he very graciously sent me this movie, and I want to show you once and for all so that you understand how alive bacteria are, what bacteria dividing look like. Buckle your seatbelts. All right? The bacterial cell cycle is 20 minutes. So one round of division, 20 minutes, 2-0. Here we go. I know. So I give you my word, the DNA is not inside the corn dog neatly packaged away. The reason this is relevant to Avery's quixotic experiment and his attempts to purify DNA is it's difficult to get rid of all the other stuff. So I'm going to walk you through the biochemical purification scheme. You don't need to know it. Let me give you a sense of scale. They started with 19 gallons of this. Just mentally visualize a gallon. Now mentally visualize 19 gallons. Now visualize 19 gallons of bacteria. So you first heat and activate the bacteria. Why? To kill them. Second, because remember, they cause lethal disease. Second, so you have 90 gallons of lethal stuff. Excellent project. You wash them with salt. Why? To remove protein, sugar, and RNA. The basic idea, this is a biochemical fractionation scheme, is you want to get rid of all the irrelevant stuff. How do you know you've gotten rid of the irrelevant stuff and what you're interested in is still there? You test what is still there for whether it's still active. And how do you test that? You add it to some benign bacteria and say, will my residual stuff transform them? If no, go back to growing at 19 more liters of bacteria. One more time. At every step in the biochemical fractionation scheme, what you still have left, you have to test using an assay for biological potency. Does that make sense? Not really? Ah, okay. You're trying to purify the transforming principle. What causes one bacterium to transform another? You take the heat-killed bacteria that have the transformation capacity and you're trying to isolate the entity that does it. So you wash it with salt. Stuff floats away, some stuff stays. Is what you're after still there? How can you tell? You can assay it. You can mix it with some normal bacteria and ask, does this stuff still transform? Yes, no. Then you wash with soap to remove protein and lipid, and with chloroform to remove the protein, and a special enzyme to destroy the sugar, then ethanol precipitate, and then repeat that 4 times for a decade. And you get from 19 gallons of bacteria 10 milligrams of transforming principle. I generated 10 milligrams of bicarbonate of soda in my lab, and I took a picture of it so that visualize 19 gallons. This is what they got after 10 years of work. Bear with me. I have 3 minutes worth, and it's worth it. First, they applied the duck test. Is this transforming principle DNA? Well, if it walks like DNA and talks like DNA, it must be DNA. So chemical composition, very much like DNA. So this entity that was left, that was potent in transformation, was chemically like DNA, but not like protein, not like lipid, not like sugar. Was resistant to proteases, enzymes that break down protein. Was resistant to RNase, you guessed it, enzymes that break down ribonucleic acid. Completely destroyed by DNase, the enzyme that breaks down DNA, and absorbs UV light like DNA. Wear sunscreen, or at least a hat. Sunlight is carcinogenic. Last but not least, and this was my penultimate slide, it was like Ice 9. Does anybody know what Ice 9 is? Kurt Vonnegut, Cat's Cradle. Read it. It's remarkable. Basically, you could dilute it down, that 10 milligrams. You could dilute it down, let's see, 10 to the minus 4, 10,000 times. Go from 10 milligrams to 10 nanograms, and it would still work. Avery concluded that when ketone-activated lethal bacteria transform normal ones, they do that by DNA. DNA is the transforming principle. Stop the presses, the cover of the New York Times. None of the above. The paper was unbelievably understated. And I have one more slide and then I'm going to let you go, I promise. Avery wrote to his brother in a private letter what he really thought, but the paper is like, well, the transforming principle is DNA. Have a nice day, I'm going to go now. This is the tone of the paper. Try to find in that complex mixture the active principle, some job full of headaches and heartbreaks. But perhaps we have it. In short, he goes, the substance conforms very closely to pure DNA. Who could have guessed it? If we are right, and of course it's not proven, I love that understatement of a scientist, it's not yet proven, then it means that nucleic acids are functionally active substances in determining the biochemical activities and specific characteristics of cells. This has been the dream of geneticists. Last slide. The Gilderoy-Lockhart phenotype. You know, the self-promotion. Stanisław Jezielec, the great Polish aphorist, wonderfully described it by saying, I'm handsome, I'm strong, I'm smart, and I'm kind, and I made all these discoveries myself. So that's one extreme. The other extreme is Chuchundra from Rikki Tikki Tavi, who sits in a corner, is afraid of anything, and never moves. Question. Think about this as you walk to your next class. Should Avery have been more forceful in the discussion section of his paper? Ask yourself, what stance will you take when you make a discovery of that magnitude? See you Friday.